There is a cell in your body unlike any other. It doesn't reproduce like the rest. It isn't easily replaced when it dies. And yet, it is the cell responsible for every thought you've ever had, every memory you hold, every movement you've made since birth. For centuries, the world's greatest anatomists peered at it directly under the microscope, unable to see what it truly was. They mistook it for a continuous network, an uninterrupted tissue, an undifferentiated mass of nerve matter. This error persisted for decades, and when it was finally corrected, it forever changed humanity's understanding of the mind. This is the neuron, and its architecture is one of the most extraordinary stories hidden within the human body. To understand why neurons matter, we must begin with a fact that is rarely stated with the clarity it deserves. The human nervous system is not simply a communication system. It is the system that makes experience possible. Without neurons, there would be no perception of pain or pleasure, no awareness of time or space, no memory. The body might have a heart, lungs, and a liver, but without neurons functioning in a network, none of those organs would be experienced by anyone. The neuron is merely the body's messenger. It is the condition of possibility for everything we call inner life. The human nervous system contains approximately 86 billion neurons. This number is repeated so often that it has lost its power to amaze, but it's worth pausing to consider. 86 billion. If you wanted to count one neuron per second without sleep, without stopping, it would take you more than 2,700 years to finish. And yet, neurons are not the most numerous part of the nervous system. Glial cells, which accompany and support neurons, outnumber them. The human brain is not just a mass of neurons. It is a complex ecosystem where neurons are the main players, but not the only ones. To understand what makes this cell so special, we must look at it closely. Its structure must be broken down piece by piece because each part has a precise function, and the way these parts relate to each other is what gives the neuron its unique ability to process and transmit information. The neuron's cell body, called the soma, is the metabolic center of the entire cell. It contains the nucleus, where the genetic material resides. It also contains the organelles that produce energy, synthesize proteins, and keep the cell alive. At first glance, the soma of a neuron appears radically different from that of other cells in the body. But what surrounds it is what changes everything. Extending from the soma are numerous, thin, branched projections called dendrites. The name comes from the Greek word for tree, and the comparison is visual and accurate. Dendrites branch out like the branches of a tree, creating a broad, three-dimensional receiving surface. Dendrites are the neuron's antennae. They are the territory where signals from other cells arrive, where information from the environment and the rest of the nervous system is received. A single neuron can have thousands of dendrites, and each dendrite can receive signals from hundreds of other neurons. The integration capacity of a single nerve cell is immense in itself, but there is one extension that distinguishes the neuron from any other cell in the body with absolute anatomical clarity. It is called the axon. The axon is a long, sometimes extraordinarily long, extension that originates from the cell body, soma, at a specialized region called the axon hillock and extends toward its destination. In some neurons of the peripheral nervous system, axons can reach lengths of over a meter. That is, a single cell with a microscopic nucleus can have an axon that runs the length of an entire leg. This is one of the most extreme structures found in animal cell biology. The axon does not conduct information in the same way that a wire conducts electricity. This distinction may seem technical, but it has profound implications for understanding how the nervous system works. The electrical signal that travels along the axon is not a continuous current flowing from one end to the other. It is a regenerated, active event at each point along the axon, rippling through the cell like a wave. This event is called an action potential. To understand the action potential, we need to take a step back and consider what separates the inside of the axon from the outside. The neuron's plasma membrane is not just a physical barrier, it is an electrochemical boundary. Under resting conditions, the inside of the neuron is negatively charged relative to the outside, primarily because there are more potassium ions inside and more sodium ions outside, and because the membrane has different permeabilities to each of them. This difference in charge is called the resting membrane potential, and in most neurons, it hovers around -70 millivolts. When a dendrite receives a sufficiently strong signal, the membrane potential in that region begins to change. If the change reaches a critical threshold, something remarkable happens. The sodium channels in the membrane suddenly open. Sodium ions that were accumulating outside the cell rush in. The interior becomes momentarily positive. This sudden change in voltage activates channels in the adjacent region of the axon, which also open, propagating the electrical wave forward. Meanwhile, potassium channels open to restore equilibrium, and the cell enters a brief refractory period before it is ready to fire again. This entire process, from the beginning of the action potential to its conclusion, occurs in less than 2 milliseconds and happens identically each time the threshold is exceeded. The neuron modulates the intensity of its signal by changing the size of the action potential. What varies is the frequency with which it fires. More stimulation means more action potentials per second, not necessarily larger action potentials. This property, known as the all or none law, has fundamental implications for how the nervous system encodes information. But the action potential needs to travel quickly for the nervous system to function. And this is where one of biology's most elegant structures comes in, the myelin sheath. Myelin is a fat-rich lipid substance that wraps the axon in segments. It doesn't coat it continuously, but rather at intervals, leaving small bare areas called nodes of Ranvier. This architecture has an extraordinary physiological consequence. The action potential doesn't have to regenerate at every point along the axon. It can jump from one node to the next in a process called saltatory conduction. This leap greatly reduces the amount of energy needed to transmit a signal, and at the same time, dramatically increases conduction velocity. In unmyelinated axons, conduction velocity can be as low as 2 meters per second. In well-myelinated axons, that velocity can reach 120 meters per second. This difference is not trivial. It is the difference between a nervous system capable of controlling rapid and precise movements and one that is slow to respond. Myelin is not an accessory. It is an evolutionary innovation that made the complexity of the vertebrate nervous system possible. Myelin production in the central nervous system is carried out by a type of glial cell called an oligodendrocyte. Each oligodendrocyte can myelinate up to 50 different axon segments simultaneously. In the peripheral nervous system, this function is performed by Schwann cells, which individually envelop each segment. When myelin is damaged, as occurs in diseases like multiple sclerosis, conduction velocity decreases drastically, action potentials are blocked, and the result is the loss of motor and sensory functions that depend on these fibers. The disease does not destroy neurons immediately. It destroys their ability to communicate with the necessary speed and reliability. When the action potential reaches the end of the axon, it enters a region of extraordinary molecular sophistication, the axon terminal or synaptic bouton. Here the neuron faces a fundamental problem. It cannot simply transfer its electrical signal to the next cell as if they were two connected wires. There is a physical gap between the axon terminal and the receiving cell. This gap, a mere 20 to 40 nanometers wide, is called the synaptic cleft. And crossing this cleft is one of the most studied and, at the same time, most fascinating processes in all of neuroscience. Inside the axon terminal are synaptic vesicles, tiny sacs filled with molecules called neurotransmitters. When the action potential reaches the terminal, it triggers an influx of calcium ions from the outside. Calcium acts as a chemical signal that causes vesicles to fuse with the terminal membrane and release their contents into the synaptic cleft. Neurotransmitters diffuse across the gap and bind to specific receptors on the postsynaptic cell membrane. What happens next depends on which neurotransmitter is released and which type of receptor receives it. Some neurotransmitters are excitatory, making it more likely that the postsynaptic cell will fire its own action potential. Others are inhibitory, making it less likely that the cell will fire. The postsynaptic neuron constantly integrates all the signals it receives, summing the excitatory ones and subtracting the inhibitory ones, and only fires if the total sum exceeds its threshold. In this process of integration, in this continuous calculation performed by each neuron at every moment, lies the computational basis of the nervous system. The best known neurotransmitters include glutamate, the main excitatory neurotransmitter of the central nervous system, GABA, the main inhibitory neurotransmitter, dopamine, involved in motivation and the reward system, serotonin, related to mood and sleep, acetylcholine, essential for neuromuscular junctions and cognitive functions, and norepinephrine, involved in the stress response and attention. Each of these messengers has specific receptors, particular synthesis pathways, and its own elimination mechanisms. Modern pharmacology operates largely through the manipulation of these molecules. Antidepressants, anesthetics, anxiolytics, antipsychotics all act by modifying synaptic signaling in some way. Neurons don't come in just one form. The morphological and functional diversity of neurons is one of the most striking features of nervous biology. Motor neurons, with their long axons, carry commands from the central nervous system to the muscles. Sensory neurons receive information from the external and internal environment and transmit it to the brain. Interneurons, which are by far the most numerous, connect with each other within the central nervous system, forming the circuits that underlie perception, thought, memory, and movement. There is a classic morphological classification that divides neurons according to the number of processes extending from the cell body, soma. Unipolar neurons have a single process that divides into two branches. Bipolar neurons have two processes, one dendritic and one axonal. Multipolar neurons, which are the most common in the human central nervous system, have multiple dendrites and a single axon. Within these categories, there is extraordinary diversity. Purkinje cells in the cerebellum, for example, have a dendritic tree of almost artistic visual complexity, branching in a single two-dimensional plane. Pyramidal cells in the cerebral cortex have a completely different morphology, with a long apical dendrite extending toward the cortical surface and multiple basal dendrites extending horizontally. There is one aspect of neuronal biology that was long considered an absolute dogma and that recent research has begun to significantly challenge. For much of the 20th century, it was taught that the adult brain did not generate new neurons. The idea was that the number of neurons we are born with is essentially the number we will have throughout our lives. This belief had apparent logic, given that neurons must maintain stable connections for memory to function. Allowing the emergence of new cells could disrupt existing circuits. However, studies conducted since the late 20th century have provided evidence that in certain regions of the adult brain, particularly the hippocampus, the generation of new neurons does occur, a process known as adult neurogenesis. Research continues to explore the extent to which this process is functionally relevant in humans, what triggers it, and how it might be modulated. There is still no full consensus on the scope of adult neurogenesis in the human brain, and the question remains a subject of active debate within the scientific community. What is well established, however, is that the adult brain maintains remarkable plasticity. Synaptic connections are not fixed structures. They strengthen when used repeatedly in a process that underlies learning and memory. They weaken when not used. New connections can form, others can be withdrawn. This synaptic plasticity, whose molecular mechanism was largely elucidated thanks to the work of Eric Kandel, who received the Nobel Prize in 2000, is the biological basis of everything we learn throughout our lives. The concept of neuronal plasticity dismantles a simplistic but persistent view of the brain as a machine of fixed circuits. The brain you have today is not identical to the one you had 10 years ago. Not only because you have lost some neurons, but because the network of connections between them has changed in subtle and profound ways. Every significant experience, every acquired skill, every consolidated memory leaves a physical imprint on the synaptic architecture of the nervous system. A question naturally arises from all this, one that neuroscientists have been formulating in various forms for decades. How is it possible that the incredibly complex activity of 86 billion cells generates something as intangible as consciousness? This question, sometimes called the hard problem of consciousness, remains open. Modern neuroscience tools allow us to record the activity of thousands of neurons simultaneously, map circuits with unprecedented precision, and identify which brain regions are active during different mental states. But the gap between describing neuronal activity and explaining subjective experience remains one of science's greatest mysteries. What can be stated with certainty is that the neuron, with all its structural and functional complexity, is the unit upon which this mystery is built. Its anatomy is not accidental. Every aspect of its morphology, from the branching of its dendrites to the molecular architecture of its synapses, has been shaped over hundreds of millions of years of evolution to enable one thing: the precise, rapid, and flexible transmission of information. The evolution of the nervous system began with simple organisms possessing only a few cells capable of responding to environmental stimuli. Over time, these cells specialized, developed longer extensions, and began to connect with each other in networks. The vertebrate nervous system, with its spinal cord, peripheral nerves, and brain, is the cumulative result of this process. And within each neuron, the fundamental principles that appeared in the first organisms with nerve cells are preserved. The potential difference across the membrane, ion channels, and the release of neurotransmitters. When the anatomy of neurons is studied in depth, something becomes evident that goes beyond the technical description. The neuron is an extraordinarily elegant solution to an extraordinarily difficult problem. The problem of how to transmit information quickly, accurately, and flexibly within a living organism. The solution involves electrochemical gradients maintained by molecular pumps that operate continuously, ion channels that open and close in fractions of a millisecond, a cellular geometry that maximizes the receptive surface area while maintaining a single exit pathway, and a chemical communication system at the synapse that allows for modularity and adaptability that no conventional electrical wire could match. The human body is full of remarkable designs. The heart that pumps tirelessly for decades, the kidney that filters blood plasma with astonishing molecular selectivity, the immune system that learns to recognize threats and remembers them. But the neuron occupies a unique place in this gallery of biological wonders. Because the neuron is not merely a technical solution to a physiological problem. It is the cell that makes it possible for the rest of these wonders to be known, felt, and remembered. It is the cell through which the human body becomes conscious of itself. And this property, that of being the physical substrate of knowledge and experience, gives the neuron a dimension that no other cell in the body possesses. Every thought you have right now, every image forming in your mind as you hear these words, is the result of action potentials propagating through myelinated axons, of neurotransmitters crossing synaptic clefts 20 nanometers wide, of neurons firing in patterns we don't yet fully understand. The inner universe of human experience has a physical basis, and that basis begins in the architecture of a single cell. If this journey through the most extraordinary architecture of the human body has left you with something new, something you hadn't seen with this clarity before, then you know the value of this kind of knowledge. In Revealed Anatomy, we continue to peel back the layers that biology hides in plain sight, with the same depth and rigor. If you want to keep discovering what the human body holds within, subscribe to the channel and leave a like. It's the simplest way for this content to reach more people.